Yes, sir.
Today's session. Okay. Okay, start to read it. Just a minute. Good afternoon to all. I welcome all the participants for this today's session. Today, our chief guest, Dr. M. Shankar, Associate Professor, Department of Chemistry, Indian Institute of Technology, Roorkee, Uttarakhand, and his topic is materials applications of porphyrin. Before we start the programs, it is our duty to tell about the convener and coordinators and the organizing committee. So now I take this time as an opportunity to tell about our conveners and coordinators as well as the organizing committee members. Aisa, please do it.
Okay, sir. Good evening to one and all. On behalf of the Department of Chemistry, B.S. Abdur Rahman, Crescent Institute of Science and Technology, I'm extremely happy to welcome you all for this fifth day FDP program on Material Science and Nanotechnology, MSNT 2020. Let's give a brief introduction about the conveners of our program. There are two conveners for this FDP MSNT 2020. They are Dr. S. Puttinani ma'am, who is the professor and dean, School of Physical Sciences at B.S. Abdurrahman, Crescent Institute of Science and Technology in the Department of Chemistry. She has completed her bachelor's, master's, and MPhil degrees in chemistry at the University of Madras. She completed her B.Ed at Annamalai University, and she began to learn MBA in Education Management at Aragappa University. Finally, she completed her PhD in Catalysis and Kinetics at Anna University. She started her career as a lecturer and in the year 1991 at the Department of Chemistry, B.S. Abdurrahman, Crescent Institute of Science and Technology, and currently she is now the Dean of School and Physical and Chemical Sciences in 2015. She has published several research articles in high impact factor national and international journals. Her research areas include catalysis and kinetics, synthesis and characterization of skiff-based complexes, and synthesis and characterization of polyoxometalates. Under her supervision, many students have completed their PhD. She is currently guiding around 12 MPhil students and 6 doctoral students. Second, our second convener is Dr. D. Ishramurthy Sir, Professor and Head, Department of Chemistry, B.S. Abdurrahman, Crescent Institute of Science and Technology. He has completed his PhD at Madurai Kamaraj University and he worked as a postdoctoral fellow at National Sun Yat-sen University, Taiwan from 1998 to July 1999. He was awarded Young Scientist Fellow by Tamil Nadu State Council for Science and Technology, Tamil Nadu. He was awarded Summer Research Fellowship by the Indian Science Academy, Brainful Scientist, and he also received Brainful Scientist Award from South Korea. His areas of research interest include coordination complexes, catalysis, and solar cell development. Under his supervision, several students have completed their PhD and he is guiding several PhD scholars. He has published several research articles in high impact factor national and international journals. And now let's see about the coordinator of this FTP MSNT 2020. It is none other than Dr. N. Vasimalai sir. He is now working as an assistant professor, selection grade at the Department of Chemistry, B.S. Abdurrahman, Crescent Institute of Science and Technology, which is deemed to be a university. He has completed his bachelor and master degrees in chemistry at Vivekananda College, Madurai, and he received the Best Student Award for both the degree period. Then he completed his MPhil and PhD degrees at Gandhigram Rural University, Dindigul, under the guidance of Professor Abraham John. During his PhD, he received UGC JRF and CSIR SRF fellowships from Government of India. After completion of PhD degree, he worked as postdoctoral fellow at the National Chen Kung University, Taiwan. Then he received globally renowned fellowship of Marie Curie Fellowship in Europe and worked at the International Iberian Nanotechnology Laboratory, Portugal from 2015 to 2018. He has published more than 30 research articles in the high impact factor international journals and 4 book chapters. One of his articles has been highlighted in Nature India. The main highlight is he is the first author or corresponding author of all his publications. Now he is guiding 4 PhD students and several master students. Moreover, he is a PhD co-supervisor of Malaysian student in University of Mara, Malaysia. He is an editor of many Journal of Chemistry, Mayfab Journal of Chemistry, Canada, and reviewer of ACS, RSC, Elsewhere, Springer, Wiley journals. He has delivered several talks in national and international institutions. His field of specialization has been focused on the synthesis and characterization of fluorescent metal and metal oxide nanoparticles, and the biosensor and biomedical applications. Recently, he has developed a technology for the 1-minute synthesis of highly fluorescent copper nanocluster and developed a smartphone-aided paper-based kit for on-site monitoring of bio and toxic chemicals. He has a research collaboration with several countries including Portugal, USA, Germany, Spain, Malaysia, Japan, Taiwan, and Australia. So with this, actually there are many things to say, but I'm just telling a short, mainly a short description. So thank you, sir, for giving me this opportunity.
Thanks a lot, Aisha. Thanks for your nice introduction about the coordinators as well as conveners.
Okay.
It is my duty to introduce my FTP students committee. Without their support, we cannot able to conduct this type of program. You know well how hard to conduct the 14 days FTP. So my students, fortunately, by God's grace, I got a very brilliant students, and they are helping in all directions. So I'm very happy to introduce my students one by one. First, research scholars. So our research scholars, first one, Aisha Begum. She only now doing the comparing. She's doing PhD under the guidance of Dr. Bhagavati. And one more senior PhD student, Dr. Yam Chandru. He's doing PhD under the guidance of our Dean Madam, Dr. S. Kutirani. And another one person, Marudu Pandi, he's doing PhD under the guidance of me, Dr. N. Vasi Malai. Then senior PhD research scholar, Mr. Harishankar, and he's doing PhD under the guidance of Dr. Revadi Purushottaman. Another research scholar, J. Immaculate Mercy, she's doing PhD under the guidance of me, Dr. N. Vasi Malai. And one more research scholar, D. Lakshmi, she's doing PhD under the guidance of Dr. Noor and Mr. S. Gokuliswaran, he's doing PhD under the guidance of me, Dr. N. Vasi Malai. So without their support, I cannot able to conduct this type of programs. And every day they are coming for the rehearsal, and every day they are helping me in all the directions during their pandemic period, as well as they are also concentrating this research. So thanks a lot, my students. In this occasion, it is my duty to thank their supervisor to giving me freedom to utilize their potential, as well as allowing me to do this type of work, especially Dr. Bhagavati and Dr. S. Kutirani and Dr. Revadi Purushottaman and Dr. Noor for their fullest support. And also I thank all the faculty members from Department of Chemistry for their direct and indirect support. And MSc, second MSc students, Mr. R. Abilas, hope you have seen the invitation video. So this person only have prepared these nice videos, very intelligent persons. Mr. R. Abilas and V. Venkadeshans, Kunasekaran and Venkatramanans, they are helping a lot, not only this team and the comparing team also, Samira, Archana, and Nishat, and Mohammed Ibrahim's, and Renita and Katisha. And then these students, these students are our second MSc chemistry students from Department of Chemistry, BS Abdur Crescent Institute of Science and Technology. This is Mr. M.A. Oscar. He's a Madras Christian College student. Now he's doing the project under me. So many people are coming from Madras Christian College. They are doing their master project at our institute, and we are allowing them to do. So take this opportunity, our institute allowing to do the 6-month project or 3 months project for the outside students. So our faculty members are helping them. So you can utilize, in case you can send your students to our Department of Chemistry to do their master project. So he is also helping me. And on behalf of Department of Chemistry, it's a great opportunity to thank Madras Christian College also. Every year they are sending lot of students. So without their support and guidance, we cannot able to do. So students also giving lot of ideas. So we are working in a team. So on behalf of this Department of Chemistry, BS Abdur Rahman Crescent Institute of Science and Technology, I also thank all of my students. Now I can stop the screen. Anna, Chief Guest Anna, you can share your screen. As I get ready to do the comparings. So it
Okay, sir.
reached 100. Okay. Screen share பண்ணுங்கண்ணா.
Yes, yes, yes.
Okay. Good afternoon to all. It is my pleasure to welcome all the participants for today's session. And today's speaker is a friend of my brother. I can also call him as Anna, Dr. Professor M. Sankar. He's associate professor working at the Department of Chemistry, Indian Institute of Technology, Roorkee. Now I request Aisha to do the comparing. Thank you.
Good evening, sir. It's a pleasure to welcome you for this fifth day of the FDP MSNT 2020. Now, let me give a short introduction about our speaker, Dr. M. Shankar, Associate Professor, Department of Chemistry, Indian Institute of Technology, Roorkee, Uttarakhand. Dr. M. Shankar earned his doctoral degree in bioinorganic chemistry from the Indian Institute of Technology, Madras in the year 2005. He worked as a research associate at Tel Aviv University, Israel, under the Israel-US binational scheme from 2005 to 2007. He was a postdoctoral fellow at the University of Bourgogne, Dijon, France, from 2007 to 2008. He was a CNRS postdoctoral fellow at the University of Rennes, France, from 2008 to 2009. He was also a JSPS fellow at the University of Tsukuba, Japan, for a period of 2 years. In 2011, he joined the IIT-Roorkee as an assistant professor, where he is an associate professor now. He has guided around 8 doctoral students and 13 master students, and currently he is guiding around 12 doctoral students. He has around 94 publications in high impact factor journals and has a citation of 1,700. He has an H index of 23 and a nitrogen index of 53. He is also a member of professional bodies such as American Chemical Society, Royal Society of Chemistry, Electrochemical Society, etc. He has become the Fellow of the Royal Society of Chemistry in the year 2020. He was a visiting professor at the University of North Texas, Denton, USA, and at the University of Houston, USA in 2019. He has received various meritorious honors and awards. He has received Outstanding Young Faculty Award with Institute Research Fellowship from IIT-Roorkee in 2018. Moreover, he is an eminent person and he has received project funding of around 140.42 lakhs as PI from BRNS, CSIR, IUSSTF, SERB, etc., and around 771.6 lakhs as a co-PI. His research works are very popular and it is even highlighted by media such as TV and magazines. His field of specialization has been focused on the synthesis and studies on porphyrinoids, coordination chemistry, supramolecular assemblies, catalysis, photophysical and photovoltaic studies, etc. With this, now I request Dr. M. Shankar sir to take over the session. Bonjour, sir.
Hello.
No, it's audible. You can.
Yeah, okay. So very good evening to all. Thanks, Aisha, for your kind introduction. My sincere thanks to the organization committee, particularly like Dr. Wasim Malay. So he has taken kind efforts to organize this event, and also Department of Chemistry of your institute. So today I'll be talking about material applications of porphyrinoids. Porphyrinoids means porphyrins and their derivatives. Simple porphyrin, that is heme. Heme has a porphyrin skeleton. So we will see in the detailed properties of these porphyrins. So I have organized my presentation. First, I will be talking about importance of functionalized porphyrins, then applications of porphyrinoids, particularly in solar cells, as well as antibacterial studies and detection of anions, particularly cyanide and fluorides, and detection of explosive, particularly picric acid, then catalytic applications using vanadilyl porphyrins, electrocatalytic applications using gobal porphyrins, and then we are preparing some NLO materials. So we will describe about that. Finally, I will conclude my presentation. As you all know that tetraprodal pigments plays important role in biological system. For example, hemoglobin does oxygen transport. It takes oxygen from lungs to tissues. Like myoglobin stores that. Like myoglobin is present in tissues, it stores oxygen. And cytochromes are involved in electron transfer reactions. This hemoglobin has a heme unit. This ring system called porphyrin. There is a tetrapyrrole pigment. It's highly conjugated. So this gives color to the blood. So the red color of the blood because of the highly pi conjugated heme or porphyrin ring. Similarly, chlorophyll plays important role in photosynthesis. They are present in thylakoid membranes. So they are the one light harvesting, like mainly they involved in light harvesting. And this is the bacteria chlorophyll. So it has a ring system called bacteria chlorine. Two of the double bonds are reduced at the opposite position. Okay, they are called bacteria chlorine. So it is involved in photosynthesis of bacteria, which help to identify how the photosystem works. Here in chlorophyll, there is only one double bond is reduced. Here, this ring system called chlorine ring system. That is green color. This because of this ring system, chlorophyll is green in color. Then vitamin B12, they are important for group transfer reaction, particularly biomethylation or synthesis of amino acids. That is the backbone for the proteins. And here you have a ring system called corine ring system, partly conjugated, cobalt metal present in it. Then cofactor F430. It has a methanocorpin ring system. This also partly conjugated. The main important role of cofactor F430 is production of methane from organic sediments. So whatever the methane it is in the earth crust, it is produced by cofactor F30. Then cytochrome P450. It's an important heme enzyme. It does catalysis, oxidative catalysis, particularly oxidation of polyaromatic compounds into phenolic compounds that were excreted through urine, detoxification process. So the diverse functions of tetrapyrrole pigments prompted the laboratory synthesis of porphyrin. Particularly heme proteins inspired the researchers to synthesize porphyrins. This is tetrapyrrole skeleton. This is called porphyrin. There is no substitution in it. There are bridged positions. 4 bridged positions are there. They are called meso positions. And 8 beta pyrrole positions are there, like alpha, beta pyrrole. So 8 such positions are there. This is an example for meso substituted porphyrin. So here you can see the alpha, the numbering goes from alpha carbon of the pyrrole in a cyclic manner. So meso positions are 5, 10, 15, 20. This is called 5, 10, 15, 20 tetraphenyl porphyrin. Then this is an example for octaethyl porphyrin, sorry, beta substituted porphyrin, octaethyl. So here, this ethyl groups are at the beta pyrrole positions. Among these two, octaethyl porphyrin is difficult to synthesize because the diethyl pyrrole is a multi-step synthesis. Whereas this porphyrin, tetraphenyl porphyrin, very easy to synthesize, widely explored. That so it has a highly pi conjugated system. So it has a discrete HOMO, that is A1U and A2U pi orbitals, and then we have a LUMO pi star EG. That so this HOMO to LUMO transition brings spectral bands, particularly like a HOMO to S2 transition. This is B band or benzenoid band, or another name is called Soret band, who described this particular transition. So here the intense band arising from around 400 to 500 nanometer called B band, that is pi to pi star, fully allowed. Then there is a partially allowed transition. They are called Q bands, that goes from HOMO to S1 of this porphyrin. Then here you will have a Q bands partially allowed pi to pi star only. So the contribution of the HOMO, like A1U, comes from alpha and beta carbon, and A2U is from the meso position as well as immunonitrogen. That so when you have electron withdrawing group at the porphyrin periphery, you can stabilize your A1U, then A1U will come down, A2U will go up. If you put electron donating groups, then you can switch it. So particularly you can tune HOMO LUMO, and then also your LUMO also will be sensitive because When you have a strong electron withdrawing group or pi extending group, your LUMO will be, pi star will be stabilized. So in that case, like you can tune the HOMO LUMO. By that you can tune this energy levels, spectral bands. That's how. So they are interesting. These are porphyrin analogs. So simple tetrapyrrole skeleton called porphyrin, then the substituted is porphyrin, as I mentioned earlier. But if it is one of the double bond is reduced, that is called chlorine. That is present in chlorophyll. Then two of the double bonds are reduced, bacteria chlorine, that is present in bacteria chlorophyll. Then this benzoporphyrin, pi extender porphyrin. These are synthetic porphyrins. And then this is meso position is missing, carol, and one of the pyrrole ring can be twisted. This is called unconfused porphyrin. Meso position can be expanded or contracted. That's called porphysine. Then here meso positions can be with sp3 carbons. That's called Gallic spherols, very famous for anion sensing. And you can replace the pyrrole ring with furon, thiophene, selenophene. They are called core modified porphyrin. And we can increase the number of pyrrole rings, like a 5 pyrrole pentapyrrole, that and 6, 7, 8 up to 12 you can go. That they are called expanded porphyrins. So today I'll mainly discuss about porphyrin and some of its analogs, like only few. Mainly porphyrins were used to mimic hemoglobin, myoglobin in the beginning. Okay, so actually that this is the picket fence porphyrin, has a pyramidal groups. So these amide groups are like gives the steric entrance to the porphyrin periphery. So when it binds with oxygen, it binds with the side-on mode. So this one only gave idea that how hemoglobin binds with oxygen to deduce Perut's mechanism. That so nowadays people are using iron porphyrins for carbon dioxide reduction, water oxidation, many other applications actually. Porphyrins also can be used as a sensors. When you have a Lewis acid metal inside, it can detect phenol, amines, thiols by axial coordination with the metal. So that will give you spectral changes one can identify. And also porphyrin can be utilized for the catalysis. Since actually cytochrome P450 does biocatalysis, synthetic porphyrins also can do catalysis. And so it has a extended pi conjugation as well as very good light absorption properties. So it was used in solar cells as a light harvesting material. So as just like mimicking photosynthesis. And they observed 13% power conversion efficiency. That means photon to power conversion efficiency was 13%. And then this is called photochlor, chlorine derivative. They used as a drug in photodynamic therapy to treat cancer. So this helps for the treatment of cancer. And this is the porphyrin having electron withdrawing group at the periphery. So when you have push-pull porphyrin, it is used in nonlinear optics, particularly when you have a two-photon absorption property that is used in bioimaging. I would like to show one example here. That here this is a blood vessel of a mice. That there actually the communication is interrupted. Then after the treatment, we can clearly see that now the communication is fine. So the two-photon absorption property gives importance for the optical imaging purpose. So the porphyrins have diverse applications. That's why people are interested to study those properties. Let us see how we can synthesize porphyrins. Actually, porphyrins are very easy to synthesize. We can take pyrrole and substituted benzaldehyde, and if you condense in propionic acid, you can get porphyrin. This is actually hydrophobic in nature, so it thrashes out in propionic acid. Just to filter it, you can get there is only one problem is that there is a side product called polypherol forms under acidic conditions. Polypherol formation was 75%. Porphyrin is around 25%. Then there is another method called Lindsay method. Actually, here this is a little bit harsh condition, but Lindsay method is a very mild condition. So first they react pyrrole and benzaldehyde in presence of TFA or BF3 etherate. Then it forms porphyrinogen. This is unconjugated one. Then when we add oxidant parachlorinil or DDQ, we can get conjugated system porphyrin. In 25 to 50% yields are better. Then other way is to prepare porphyrin is McDonald method. Here you can take dipyramidine dialdehyde condense with another dipyromethane, and then you oxidize it, you will get porphyrin. Other method is that you can prepare dipyromethane of having different substituents, condense. This is called billane. When you condense the billane further, then you can get porphyrin. If you take heme, if you do retrosynthesis, you will get billane. The same way you can prepare porphyrins. So these are the methodology for meso-substituted porphyrins. If you want to have a beta-pyrrole substituted porphyrins, then you have to take a porphyrin, then you can do the functionalization. This is called post-functionalization. That or you reduce the ring, then you can get chlorine, then you can use it for functionalization, or you can annulate beta position with a misophenyl. We can prepare pi extended porphyrin by annulation. Other method is called pyrrole modified porphyrins. Then you can break the pyrrole ring, and then you can do modifications. In our lab, we generally like work on beta functionalized porphyrin post functionalization, and then preparation of chlorines and fusion. These three methods. And here we mainly do core functionalization. That means beta pyrrole and misophenyl directly linked. That is actually better because they are in conjugation with the porphyrin pi system, so that the effects will be more. And also, like by doing different substitution, we can tune the porphyrin planarity. Like we know that oxyhemoglobin is planar in structure, that they are planar, but deoxyhemoglobin used to have a domed structure, dome. And then if you have a substitution at the porphyrin periphery, it can also have a wave distortion. Then it can also have a ruffled distortion. Then finally is the saddle shape conformation. This saddle shape conformation, when you have a highly sterically hindered groups around the porphyrin periphery, and it shows saddle distortion, that two pyrrole rings are up and two pyrrole rings are down, so that it minimizes the non-planarity. Saddle shape conformation is this is the one. Like so, it can adopt different conformations, depends on the sterics. So these are the four commonly known. That that's these are. We are also tuning this, because when you tune this, what happens that actually it interrupts the pi conjugation. Your HOMO rises, then HOMO-LUMO gap also decreases. Then you can expect redshift in the absorption spectral features. That so actually in our lab we synthesize porphyrins, chlorines, corals, mainly beta functionalized, sometimes meso functionalized, and then utilize them for reduction of toxic ions, and you use them for catalysis, electrocatalysis, dye-sensitized solar cells, and we use porphyrin pi system to interact with the fullerene, because fullerenes are highly electron deficient. So we can have a pi to pi star transition, pi interaction, and we can prepare post-gas assemblies based on that. So now I'll discuss some of the applications what we are doing. First one is the solar cell, followed by like photodynamic therapy, sensing, catalysis, nonlinear optics. Like first one is the solar cell. So we know about solar cells actually. So everywhere, like we install solar systems. Okay, so it's very important. So India has a cumulative solar system installation in 2018 is about 6.5%. Then in 2020 we improved by 3.2%, 9.8. So our government encourages to install so much of solar systems. So that will be helpful to get renewable energy sources. So you can see that in 2 years we have a tremendous improvement, like utilizing solar power. So why solar power? Actually, solar power is eco-friendly and clean, and also like it saves our money, because that solar power is very much abundant. Okay, so just you have to harvest them. Then it requires low maintenance. Just once in a while you need to wash with the water the panel, so that dust and all it will move off. Then it has a diverse application, so you can use it for various applications. That So actually, this solar cells, it's interest of photosynthesis actually. So we are mimicking photosynthesis. So here chlorophyll is the light absorber. Then photosynthesis, the production of glucose, uh, like so using carbon dioxide, water, chlorophyll, and light. So one can produce glucose. That's what we learn from photosynthesis. So we are going to we can mimic using porphyrins also. Like before that, uh, I would like to mention that this is a recent development in March 2020. So there was a largest solar power system installed in Badla, Rajasthan, about 2,250 megawatt project. So 10% of the state power goes from this project. And also, you know that Cochin International Airport also is operated by solar power. This is the first fully solar powered airport. So about 30 megawatts power they are getting. And also they got the Champions of Earth award from United Nations. There. So here the solar cell based on silicon is a PN junction. Okay, when you shine light, so there is a photoelectric effect. Then that you will have a exciton formation. Then that means that electron hole pair. Then movement of charges. So that from that you collect electrons. That is electricity. That so you should have appropriate electrodes. So here, this is the photovoltaic effect, as I mentioned earlier. That here, so movement of hole is a uphill process. Movement of electron is a downhill process. Please move this window away from what it means like.
It is not from our side, sir, actually. It is
Okay.
from- yeah.
So actually, as far as solar cells considered, like there are three generations. First generation is the monocrystalline silicon and polycrystalline silicon. Okay. Then second generation is amorphous thin film. Okay. Actually, first generation solar cells were very expensive. That you need 99.99% purity of silicon. Okay, 99.99% silicon is needed. So that's very expensive. But later they used amorphous thin film of silicon that reduces the cost. Now third generation solar cell is much cheaper. Okay, they are called like dye sensitized solar cells that I am going to explain. Then quantum dot sensitized solar cells, organic photovoltaics, polymer hybrid solar cells, perovskite solar cells. Perovskite solar cells also is a hot topic nowadays. Let me move on to dye sensitized solar cells. So dye-sensitized solar cell was developed by Professor Michael Gratzel. Okay, so he is working on this topic since 1972. He constructed using chlorophyll and zinc oxide electrode. Actually, this DSSC has four important components. One is titanium dioxide, that is the semiconductor, and then the electrolyte, redox couple, iodide, triiodide, and cathode and photosensitizer. So the operation principle is like this. So you have to coat your sensitizer on the ITO plate. Before that, you have to coat TiO2. So first step is that first you have to take ITO plate and then coat TiO2 on that. Then after that, you dip into the porphyrin solution or any dye solution. Dye will absorb. You excess dyes you can wash off. Then it will form a mono layer. Okay. Then like when you shine light on that, so then the dye absorbed on that, it will go to the excited state. There, so you will have excited state dye that will undergo oxidation. So the electron will come out. Then TiO2 will take up because conduction moment of TiO2 is lower than the excited state of dye. So the electron immediately will come to TiO2 and it will swim through the nanoporous TiO2. It will come out. Then you can take the load, and some electrons can be allowed to go to cathode. There actually you have a electrolyte. That electrolyte first will get reduced then followed by oxidation. You can see here triiodide get reduced to iodide, then it will get reoxidized. So that oxidized electron will come and regenerate the dye. So as long as when you shine light, the dye will go to the excited state, it will give electrons that will be reduced by electrolyte. So this process keep going. So then you can produce electricity. That's what in solar cells in the street and all. The upper part is the solar cell, down is the battery. So whenever that solar system works, it is getting charged. So actually, there are different dyes were used for solar cells. Particularly in the beginning, they used ruthenium-based dyes because it has a intramolecular charge answer. Okay, so MICT was there. Then it can be used, but the problem with the ruthenium dye is cost is very high, and also they are phototoxic. When you goes to water system and light falls on that, then it is phototoxic. So they nowadays it was stopped using this, and porphyrins are best dyes because they are biocompatible, naturally occurring, so one can use widely. So there is no toxic effect in water. They were widely used. So and organic dyes also used because organic dyes actually it doesn't go absorption in the visible region, particularly 500 to 800 region. But people also interested. They have developed several organic dyes. They also used for decentralized solar cells. Actually, we started this research in 2013 after establishing our lab. Then when I look at the literature, these porphyrins, whatever they used for decentralized solar cells, they are synthesizing in 15 steps or minimum 12 steps they need to have, they need to synthesize, they need to do. So I thought that why don't we shorten the methodology in 2 or 3 steps that I will explain the next slide. That the main thing is that they have prepared a porphyrin and append groups. Okay, so one by one using palladium catalyzed methodology. I thought that palladium catalyzed methodology cannot be used in industries, like it will be expensive. Thought they have prepared push-pull porphyrins having pushing group here, electron withdrawing group. So we also want to have a push-pull group because that will give you good electron injection. Recently also Imamuri et al. They have prepared the dye having 10.7% efficiency, but one has to synthesize in 12 steps. But we designed push-pull porphyrins. So here we have a donor group. Donor group ranges from phenyl to phenothiazine, and then we have 2 mesithyl groups. The mesithyl groups is needed because otherwise in monolayer porphyrin will aggregate. So this will prevent the aggregation, mesithyl group. Then carboxyl group will have a binding with the TiO2, that is good. So we designed this, and then synthesis is very simple. We have taken mesithyl dipyromethane. One can prepare gram quantities by condensing pyrrole and mesitoldehyde in quantitative yield. Then you condense the 2 different aldehydes in presence of TFA. We isolated this porphyrin, and ester hydrolysis gives acid porphyrin, followed by zinc insertion that gives our dye. This is only 2 steps, easy to prepare. You can get very good yield. Then we have done the photovoltaic studies. You can see here when you take phenyl, it's a simple phenyl groups, and when you take phenothiazine, carbazole, triphenylamine, the N-dimethyl phenyl, bisthioneal, they are very good donor groups. So they can donate electron density to the porphyrin pi system, so that we had a very good photon to current conversion efficiency. We have to look at other parameters also, that VOC, that and short circuit current JSC, this open circuit voltage. So this also increases. That when we have a better donor, these parameters also increases. That's why the phenothiazine appended dye gives 7% efficiency. It's very good, and synthesis is very simple. That then later on we synthesized triphenothiazine substituted porphyrin. It showed 7.3% efficiency. Then we tuned to dyes, phenothiazine dicarboxy. We had a solubility problem. We observed 5.5%. Later, we co-sensitized with N71 dye, that is ruthenium dye. we improved the efficiency 8.8. The reason is that ruthenium dye absorbs around 500 nanometer. Our porphyrin dye does not. So if you combine a dye which absorbs 500 around 700 to 800, then really you can increase the efficiency. That way we can improve the efficiency. We are working on those aspects, and like nowadays porphyrins are also used as a whole transporting layer in perovskite solar cells. We are working in this direction also. So with this, I'm like finishing my solar cells work. Like we are able to prepare efficient solar cells. Then I'll move on to other topics like catalysis and other parts. Like here, one example I would like to show is that this is the decentralized solar cells installed on Geneva airport. So you can see that they are using for electricity production, and also there are e-filling stations that DSSCs were used for charging vehicles in the bus stops. This is in Switzerland. That so the decentralized solar cell has a potential. One can use it. That people are currently working on that. So now I'll move on to photodynamic therapy. So photodynamic therapy, you must be knowing, using light. Okay, you can do the cancer treatment. So porphyrins, since having very good light absorption property, you can use it in photodynamic therapy. That so photodynamic therapy is nothing but, so you like these are the cancer cells in our like in the body. Then actually you inject porphyrin. That porphyrins are absorbed by this cancerous cells, because cancerous cells pH is slightly lower, and it can have a very good absorption of porphyrins. So porphyrin binds to that. Then after that you irradiate with the light, so that it produces singlet oxygen. Then that singlet oxygen kills the cancer cells. So here you can see the laser guided surgery, that laser lights is shined on the parts, like okay. That so here is the mechanism. Like what happens, that porphyrin absorbs the light, so it goes to the excited state. In the excited state, like, this is singlet state. From singlet to it goes to triplet state. That in the triplet state, that it transfers energy to triplet oxygen, it generates singlet oxygen. So singlet oxygen is highly reactive. It oxidizes the cancerous cells. So in that, eventual death of cancer cells takes place. That this is one type of mechanism. Other type is that it can undergo electron transfer, so that it can transfer its electron. That in that case you can have hydroxy radicals. Other radicals can form. The radicals are very reactive. They destroy the cell. So either way, singlet oxygen production or radical generation, we can kill the cancer cells. So what are the good what are the requirements for an efficient photodynamic therapy agents? Like it should be selective towards the defected cells, and therapeutic window is 700 to 950. You can think that visible region is 380 to 780, or like, but here whenever we deal with this intravenous system, blood is there. Blood has intense absorption till 630 nanometer. So blood also can absorb the light. To avoid that, therapeutic window is considered from 700 to 950, so that blood will not absorb light. And also it should have a high singlet oxygen quantum unit. So if it can produce high amount of singlet oxygen, then you can efficiently kill cancer cells. Of course, the drug should be water soluble. Then we can inject intravenously. That. So here, this porphyrinoid, particularly this is chlorine. That the double bond is reduced, but name is called a photochlor. It is widely used for anti-cancer therapy. So here you can see that IF and cat was infected. So after treatment, it was cured. Then horse hide. Okay. So here also cancer cells were cured. Then it is very good for skin cancer. That, and then esophagus cancer also can be cured. So it's a very good technique, non-invasive, that one can widely use it. So in this direction, we also worked on use this photodynamic therapy for antibacterial studies. Like this, just this is our multi-valve carbon nanotube appended porphyrin appended system. This we would like to use as a PDT drug for this uh, antibacterial activity. So before that, I would like to mention there are so many FDA approved drugs are there, porphyrin based for photodynamic therapy. Particularly this photochlore is available US, Europe, Japan, many countries, they use this photochlore as a photodynamic agent. Then these are the commercially available drugs for photodynamic therapy. So our interest is that to prepare these drugs in a simplified manner, or PTT agents in a simplified manner. We have taken pyrrole, benzaldehyde and nitrobenzaldehyde. We condensed, we isolated mononitrotriphenyl porphyrin, then reduced, we got monoaminoporphyrin. Then we have taken SWCNT because carbon nanotubes have very high surface area, good electron conductivity, uh, and also this high thermochemical stability. So that we oxidize this SWCNT with nitric acid, it forms monocarboxy nitric acid. Then we condense with aminoporphyrin. That so when we condense with aminoporphyrin, like this amide linkages, then we got porphyrin appended SWCNT. So we did absorption spectral measurements. When we measure absorption spectral measurement, SWCNT does not have absorption visible region, but in solution porphyrin is intense. But in case of nanocomposite, it's less, but it's still porphyrin is there, which indicating the Soret band, and also it showed feeble emission, so that indicates that there is a porphyrin is there, because SWCNT does not show any emission, and in solution it shows intense emission. Then further we did FESEM analysis. Like you can hear this is the carboxy appended SWCNT. When you append porphyrins, morphology changes, and this is the TAME image of carbon nanotube. You can see this tubular structure. Then this nodes on the tubular structure indicates that porphyrin appended on that, and also here we did the thermochemical studies. So this is the SWCNT COH. It has a very good stability till 500, but after appending porphyrin, slightly decreases, but still it is stable till 400 degrees Celsius. Slightly decreases because porphyrin is a carbocyclic macrocyclic system. They may lose at higher temperatures. That so we did antibacterial studies, like we used porphyrin SWCNT conjugate, and that we had a bacterial growth, like here SRS bacteria has been grown. Then when we keep this porphyrin that with the bacteria like the colonies, then the bacteria won't grow when the porphyrin was there, that under light actually. So that this is the light halogen tungsten lamp was used. In absence of light, it grows. Okay, so this is the untreated bacteria. You can see a lot of bacterial growth are there. Then after treating with the light, then as I mentioned, photodynamic activity is there. So it kills all the bacteria. When wherever the porphyrin is there, in presence of light, you didn't see any bacterial growth. If there is no exposure of light, porphyrin was not there, then there is a bacterial growth. Then we can see here also, this is the FESM of this SRS untreated. Then we treated the like for 10 minutes. This is for 30 minutes. This is for 60 minutes. In 60 minutes, you can see completely there is no bacteria, that completely it has been killed by the porphyrin, that possibly it activates singlet oxygen. Then you can see this that survivability of cell at zero concentration when there is no porphyrin, 100%. When we increase the concentration of porphyrin very less, 0.08 milligram per ml, it completely kills the bacteria. And also we did that time dependent measurements. Then in the beginning it was 100% survival. Then after 60 minutes with 0.08 milligram per ml, completely destroys the porphyrin. So very small amount is needed when porphyrin appended on SWCNT for antibacterial activity. So it is a very good result that if simply you take MWCNT, it doesn't do any photodynamic activity. So porphyrin is important. It is used as a photodynamic agent. Now I'll move on to other topic. We are interested in sensing. So sensing is very important because cyanide is widely used in many industries, uh, particularly plastic industries, as well as gold mining, and as a synthetic precursors in synthetic fibers, many more applications. Uh, and fluoride also, after particular level, it causes fluorescence. So the identification of fluoride and cyanide is very important in water systems. And also the picric acid. Picric acid is one of the most widely used explosive. So we need to have a good sensor to detect explosives. Actually, if you consider porphyrins are very good molecules to act as a sensors because it has a rich spectroscopic as well as redox properties. Porphyrin can undergo oxidation as well as reduction, and it has a very good intense absorption in the visible region. So it is very sensitive to the analytes. Then it can change its color, can be used as a colorimetric sensor. So you don't need to use any instrument to detect analytes. So you can do by naked eyes without having any instrumentation. That is the advantage of porphyrins. I'll show some examples for that. These are the literature known cyanide sensors. So you have to do multi-step synthesis and difficult to prepare, and that reduction limit is also not so good. So these are the essentials for anion sensor. It should be easy to synthesize. We will show that our sensors are easy to synthesize. We can detect by naked eyes, no need of any instrumentation, high chemoselectivity, low detection limit, and many of our sensors are recyclable. You can recycle and use it for a number of times, and we can detect in water samples also. We can detect in water. So this is the first example I am taking. Here, the porphyrin having one nitro group and 7 bromo groups. As I mentioned earlier, when you have that more number of substituents at the periphery, so it will have a non-planar conformation. That means 2 pyrroles will be up and 2 pyrroles will be down. So in that case, that NH of the porphyrin is exposed. So when we treat with the different anions, then it shows color change, particularly fluoride, acetate, dihydrogen phosphate, and cyanide, because they can have hydrogen bonding with the porphyrin pi system. That's what happening. So when you add anion, they interact hydrogen bond, then slowly deprotonates porphyrin. So you see the color change, that porphyrin is green in color. Generally porphyrins are like purple in color. When you highly substitute, they are green in color. After that, when we add anion, this is that anionic porphyrin, that is pink in color. So we add anions, it goes to pink. Then when you add small amount of trifluoroacetic acid, it regenerates back. Then we can use it for several cycles. The reduction limit was nanomolar. This is the lowest reduction limit so far known for this non-planar porphyrins. Then actually we were worried that like it doesn't show any selectivity. It is showing the same color for fluoride, acetate, dihydrogen phosphate, and cyanide. Then what we have done is that we have protected the core. So here we appended nickel. So when nickel is there, so the core is now protected, and here we kept 4 cyano groups. They are electron withdrawing in nature, so that the metal center also will be electron deficient, ring also will be electron deficient. So the metal will bind with anion. So our idea was true. It binds selectively with cyanide because cyanide pK is 9.6. Then it's a basic anion, good nucleophile. It nicely interacts with the porphyrin nickel. then you see a red shift, like oxyhemoglobin or cytochrome P450. When it binds axially, you will see a red shift. Then we did the spectral titration. We observed that that is 1:2 stoichiometry according to the Jobs plot, and then this it showed very high binding constant. Generally, nickel tetraphenyl porphyrin without any substitution does not show any binding because it's a stable square planar complex, doesn't bind with anion. Since we made them electron deficient, it showed very high binding, very low deduction limit, 0.13 sub ppm. It's very good deduction limit. That so it is also reversible. When you add cyanide, it shows sensing. Then when you add small TFA, it regenerates back. You can use it for numerous cycles as long as you want. And we made a test strips. Test strips are very easy to make. You take a Wattman filter paper and take the dichloromethane solution of porphyrin. You dip into that and take out, dry it for 2 minutes. Your test papers will be ready. This test paper is like pH paper. So you can dip in aqueous as well as non-aqueous solution of different analytes. So when we dip, it showed color change, sharp color change with cyanide ion, green to yellow. And not only cyanoporphyrin, the having bromo with the nitro. Now, earlier I mentioned that is free base without any metal. Here when you have metal, it selectively shows cyanide. So the bottom line is that if you have a reduction potential of minus 0.8 volt or positive than that, that is considered as electron deficient, that will bind with cyanide ion. It can be used as a sensor. Then another trial we made is that like if you take electron rich porphyrin, then having dicyanovinyl moiety, when you add cyanide ion, the cyanide ion will not bind with the nickel because this electron rich porphyrin, it will bind with the periphery. That's what we have observed. We have observed a blue shift from 450 nanometer to 460 nanometer. When we do the cyanide titration, the solid band decreases and new band rises at 460 nanometer. Then it is showing colorimetric change. Even you can use it in water, and we did the test strips. That's also detect cyanide ion. Even we use the TLC plate to detect cyanide ion. So you take porphyrin and add all the analytes. When you add cyanide, it shows different RF as well as color. That so it showed ratiometric sensing that in presence of 100 equivalents of these anions, still it detects cyanide ion. Then the mechanism is like this. This cyanide ion is a good nucleophile, attacks the activated double bond, uh, basic in nature. So it forms tricyanoethyl derivative that was proved by NMR spectroscopy. So not only like we prepared this dicyanovinyl, we prepared with ethyl cyanoacetate. Still it works as a cyanide sensor. We say as a chemodosimeter because quantitatively you can detect cyanide ion. That is chemodosimeter. Then when we change the linker, that is cyanoacetic acid. So acetic acid, you know that acetic COH proton can be easily deprotonated by fluoride cyanide acetate. It acts as a chemosensor. Here we placed phenyl groups in between. So when just cyanide sensing, but spectroscopically we are not able to observe. So that means that we can porphyrin pi system is important. Then it should be in conjugation with this acceptor group so that it can have a color change. The detection limit is much lower than cyanide sensors. Sorry, cyanide derivatives. It is 0.023. It's very good. It can detect very low amount of cyanide. Then actually we developed our sensing strategy. Generally like one sensor, one this analyte. It is time consuming and also each time you have to prepare sensor. Then what we thought this, we want to approach, we want to use the methodology called Lebonia molecule approach. One molecule can detect many analytes. So in that approach, so we prepared this porphyrin. It's a very simple porphyrin. If you remember earlier, I used dicyanovinyl. Now we changed to this simple phenyl to 3,5-ditertiary para-hydroxyphenyl. So in that case, so here you have hydroxy group. When it hydrogen bonds with the fluoride ion, so anion generated, then it get oxidized. This is called phosphodimethyl, blue in color. Then when you add cyanide ion, it detects here tricyanoethyl. Then it can detect cyanide fluoride simultaneously. Sorry. Here, when we don't have any metal here, when we add fluoride ion, it completely get oxidized. This is called porphyrinogen. Porphyrinogen. Then their fluoride binds. After that, water wash, you can get porphyrinogen, ascorbic acid. That is vitamin B12. If you add, that is acting as a reductant that regenerates porphyrin. Like that, you can generate back your porphyrin. If you add cyanide ion, it reacts here. So, like you use here porphyrin and deduct fluoride ion, do water wash, excessive water wash, get back your porphyrin. You can use it for several cycles. Here also you can take free base uh, porphyrin, then you can deduct fluoride ion and regenerate back. So like something like writing, erasing, volatile memory. Then what we have done is that we have taken porphyrinogen, we appended dicyanovinyl moiety. So that selectively detects picric acid among all nitroaromatics. Because picric acid is highly acidic in nature, this has a NH protons, it protonates, it generates picrates, that is hydrogen bonds with the protonated porphyrin. It shows different color, maroon color. So we can detect up to 1 ppm. Then not only picric acid can detect fluoride ion as well as cyanide ion simultaneously, like it interacts with the porphyrin pi system in a different manner, so that simultaneously you can detect these three analytes. This is an first example. It can detect three analytes in a very good manner. Then we prepared boronic ester appended porphyrin. They are very simple. Okay, you have boronic esters. When you add anion, it forms a boronate salt. Then when you add neopentyl glycol, it regenerates back. Let us see here that you have a zinc porphyrin that absorbs around 450 nanometer. Then when you add cyanide ion, it goes to 440, green in color. Then fluoride ion around 430, dihydrogen phosphate around 420. Then when you add water, it precipitates. When you add neopentyl glycol, it regenerates back. So we can regenerate our sensor after sensing. So far we have discussed several strategy to synthesize sensors, and they are also efficient for cyanide, fluoride, picric acid and all. Now I'll move on to catalytic applications of porphyrins. Actually, porphyrins are very good ligands, and with vanadium metal, so it can act as a robust catalyst. Generally, people are working with the planar porphyrins. The problem with the planar porphyrins is that it can form mu oxodimer that way can become catalytically inactive, or like sometimes it can undergo ring opening reaction. When it ring opens, then it is catalytically inactive. Uh, so here we have prepared octochloroporphyrin having chloro groups at the periphery. It gives robust structure to the porphyrin pi system. Very good. So we used for epoxidation reaction very low amount of catalyst, and then sodium carbonate as a promoter in acetonitrile water mixture. We selectively got epoxide. Then we used the strategy for several olefin. We got very good selectivity and very good top number. The top number is from 6,500 to 9,500 is very good. And we isolated the intermediate. This is oxoperoxide intermediate. This is identified by NMR and also mass spectrometry. This is the responsible species for epoxidation. Then we double the efficiency by appending 16 bromo groups at the porphyrin periphery, 8 at the beta ferrol and 8 at the mesophenyl groups. Then it's a robust catalyst that the efficiency is doubled. Then selectively does epoxidation, and further we use them for oxidative bromination of thymol. Thymol is a phenolic compound. Then we brominate thymol because thymol is an antiseptic agent industrially used, and bromothymol increases its activity and lipophilicity, so that it has an industrial importance. And recently what we have done is that we have taken vanadilyl porphyrin and we did not use any phenolic derivatives. In absence of that, under bromination condition, it forms octabromo derivative, 96% yield, isolated yield. Then we can use it for olefin epoxidation as well as bromophenol, this oxidative bromination of phenols. Like here, this is also oxoperox intermediate. It reacts with the bromodione, forms the bromonium intermediate. That is the responsible for bromination. So that we can able to prepare very efficient catalyst for epoxidation and oxidative bromination. I would like to show one example for electrocatalysis. You know, multi-wall carbon nanotubes, they have a large surface area, high conductivity, and very good thermal stability. So we can append porphyrins on that and then use it for electrocatalysis. So electrocatalysis is important, like particularly fuel cells. They are actually you can generate electricity using fuel cells. So here what we want to do is that oxygen reduction reaction. So the reduction reaction is cathodic reaction, and we can prepare cathode material. So oxygen reduction is like oxygen reacts with water that with the 4 electron reduction, it forms hydroxide ion in alkaline medium, or oxygen with reacts with 4 proton, 4 electron, it forms water in acidic medium. So here sorry. So we have taken MWCNT, we added nitric acid, set for 6 hours, we can get MWCNT with COOH. Then we added porphyrins, it absorbed on the porphyrins that and we put it on the electrode surface. So we immobilized on glassy carbon electrode, use it for oxygen reduction. When you use it for oxygen reduction, it selectively converts to water. Here we used carboxy, hydroxy, and without any substitution, tetraphenyl porphyrin. This carboxy will have a very good hydrogen bonding with COOH, complementary hydrogen bonding. The hydroxy also will have hydrogen bonding. Hydroxy porphyrins are easy to oxidize, so I think it will show better efficiency. That's what we have also observed. Here we characterize this material by various microscopic technique as well as spectroscopic technique. Here you can see that TEM, the nodular structure, when you append porphyrins, there is a this block linings are there that indicates that porphyrin appended on. That also shows cobalt metals was there, and then we characterize by XRD, that TGA as well as surface area. So simple MWCNT will have a lower surface, sorry, higher surface area. After appending porphyrins, the surface area will decrease. So we characterize by many techniques. Then we use it for oxygen reduction. So here the cobalt can get oxidized, cobalt 2 to 3, that will transfer electron to oxygen. Then oxygen reduction takes place. Or otherwise we can bring cobalt 2 to cobalt 1, and that can react with oxygen. That will convert cobalt 1 to 2. Then that way that will reduce oxygen both ways. So these catalysts are very efficient. Up to 3000 cycles, there is no shift in potential. Very good. But commercially available platinum carbon, it shifts its potential and reactivity decreases. And we did methanol poisoning studies. Our catalyst, particularly hydroxy substituted catalyst, is very stable because it has a very good hydrogen bonding as well as electron donating group. So it can stabilize the cobalt oxidation states. But then later carboxylic acid decreases slightly, then without any substitution. The classic carbon is very low, it decreases very much. So that modified electrodes has a good tolerance, methanol tolerance, can be used for oxygen reduction reaction. The last example I would like to show is that NLO, particularly push-pull porphyrins are good material for NLO activity. So here pushing group is triphenylamine, then pulling group is nitro or CHO. So this is a push-pull porphyrin. It has a very good dipole moment, 7.66. When you don't have any substitution, hardly shows any dipole moment. We studied their nonlinear optical properties using Z-scan technique. You can see here nitro substituted porphyrin shows higher nonlinearity. Then recently, like we developed tetracyanobutydiene appended porphyrin. It's a very good group. There is a charge transfer between porphyrin and the acceptor, and we did the nonlinear optical studies. It showed good nonlinearity. When we compare this TPA appended porphyrin, here we use higher laser power, so there is a thermal nonlinearity is there, but here we use lower kilohertz laser power. So kilohertz laser power mainly is their true nonlinear properties. They are very good, so we are exploring their property. And also we did optical limiting studies. We have taken porphyrin solution we shine laser light, porphyrin solution absorbs 75% of laser. That means that it can use it for the coating on spectacles. So it can absorb laser light for 75%. That means that it can be used as a glass protector, laser protector, that there is a very good optical threshold. So I would like to summarize my presentation. We could able to demonstrate simple, efficient, stable, cost-effective sensitizers for dye-sensitized solar cells. We are also able to tune spectral and redox properties of the porphyrin pi system and utilized for sensing applications. Particularly, we utilized electron deficient porphyrins for cyanide, fluoride sensing, as well as picric acid. We have synthesized robust non-planar porphyrins for epoxidation as well as oxidative bromination. Cobalt decorated MWCNTs were useful for the oxygen reduction reaction. That is the cathodic part of fuel cells. And now we are in the process of constructing model fuel cells and use it for applications. And so we are also developing good NLO materials for two photon absorption and optical imaging. And like we would like to work on porphyrin based catalysts and also porphyrin like utilization of porphyrins for extended fullerene inclusion. So gas complexes with porphyrin fullerene for photovoltaic applications, and also Carroll's derivatives are very good for catalysis and electrocatalysis. And we also have quite that interest in preparing pi extended porphyrins because they absorb in NAR region, which can be used for photodynamic therapy, anti-cancer treatment. And also, like we have prepared low amount of sensors, so we would like to use it for real sample applications like industrial effluents, sensing of explosives. One project we are working on removal of sulfur from diesel. Actually, diesel has a refractive sulfur compound called dibenzothiophene. Then we are trying to remove that dibenzothiophene by oxidation using cobalt porphyrin. Dibenzothiophene will convert it into sulfone that can be extracted in polar medium. So our porphyrins are very good. We are able to convert 96%, but temperature is slightly higher, about 60 degrees. We are trying to use modified porphyrin so that room temperature this reaction can occur. Actually, I am very much thankful to my students. Currently, like many students are working in our group, so they are putting very like hard work, and they are developing several systems for various applications like the solar cells, enhanced sensing, NLO, and wide variety of projects they are working. I also fortunate to have very good students. Like so far, 8 students have been graduated from my group. First students was Dr. Ravi Kumar. Now he is the faculty in NIT Srinagar, and Dr. Nivedita Chaudhary, she worked on fused porphyrin. All the sensing work is done by Dr. Mandeep. Now she is in National Institute of Material Science. This is Nitika, she worked on push-pull porphyrin. Solar cell work was done by Kamal, and Pinky, she worked on some extent of sensing and corals, and Tausif worked on catalysis. Pinky worked on NLO material. That I have fruitful collaborator, Professor Maurya for the catalysis, Professor Eswira for NLO, Carl Kadish for structural chemistry, Professor Krishna for DSSC, and V. Ganesan electrocatalysis. We mainly synthesize and characterize, and we do some of the studies. Then some of the studies we approach collaborators to complete. Then I am thankful to faculty staff, Department of Chemistry, IIT-Roorkee, and also thankful for the funding agencies, SCRB, CSAR, BRNS, Indo Science and Technology Forum, and IIT-Roorkee for infrastructure. And thank you for your kind attention. Thanks.
Thanks a lot for your excellent talk. No? So very nice talk, and it is very
Thank you,
informative and very useful things you just discussed. Definitely, it will be useful for our faculty members, research scholars, and students. No?
Thank you so much.
Now I give the opportunity to the participant. You just unmute your mic and talk with our chief guest. Those who are in Zoom, so you have opportunity. So please use that opportunity. So we are providing the opportunity to talk directly with our chief guest. Unmute your mic, you can ask.
Please feel free to ask questions. Uh, I like to know your.
Someone talking? Yes, madam. Vengat or Ghost, if anonymous noise is coming, please unmute mute their mic. Okay, now
Okay.
we have received the question from chatting box of Zoom as well as YouTube. So if you don't mind, can I ask that question?
Yes, please. Yes, please. Yes, Osman.
Okay. So before I go for the question session, I would like to say something about our today chief guest. So Mr. Dr. Rajiv Kondi is my best friend. Me and Dr. Rajiv Kondi, we done PhD at Gandhigram Rural University, Dindigul, and he's also was my roommate when we are in the hostel. So very nice person, and he's now working as a scientist in Bharat Petroleum. His elder brother only today chief guest, Professor M. Sankar. So that's why, because of that love and affection, I used to call him as a Sankar, sorry, as a Anna. So Anna is a very nice person, and also I know very well Anna just grown up from very small remote village from Tharmaburi district, and he done a lot of hard work. Now he's a associate professor at the Department of Chemistry, IIT-Roorkee. Happy to see you in that position now. And on behalf of Department of Chemistry, BS Abdur and Crescent Institute of Science and Technology, on behalf of our FDP participants and myself, thanks a lot. Now I can go for the uh, question from chatting box. Dr. Mehre sir asked about can we able to synthesize heme porphyrin? Heme porphyrin.
Actually, yes, one can synthesize heme. There is no problem, but it is a multi-step synthesis. Okay, so my suggestion is that you extract heme from blood. Okay, that will be a better way you can get it. Or commercially available. Heme is commercially available. You can get it. So synthesis of heme, it will be multi, because heme has unsymmetrical substituted porphyrin. Okay, so you need to do multi-step synthesis. It is possible to synthesize.
Okay, Harishankar and co-host, please check the participant's
Muted, sir.
mic. If
Muted, sir.
anonymously mute,
Mute.
unmute, please mute it.
Muted,
It's
sir.
disturbing us, yes. And another one question from Pallavi Panavan. So from Dr. Ambedkar College. Sir,
Okay.
can we able to synthesis porphyrin nanoparticles?
Yes.
Porphyrin and metal
Yeah, very good
compound
question.
nanoparticles.
We are working. Actually, currently, one of my students is deputed for porphyrin nanoparticle. Okay, so we could able to prepare porphyrin nanoparticle up to 50 nanometer, 70 nanometer size, even 30, 40 nanometer also possible. We are working on that. We would like to use it for photodynamic therapy, many other applications, electrocatalysis. We are working on that. It is possible. Reports are there.
Okay, thank you, no. So now again, I am requesting the participant. You just unmute and you can ask directly to the question to the our chief guest. Otherwise, you just raise your hand and our co-host will unmute your mic. Vengat, please check anybody is raising hand.
Nigat Nazir, please unmute yourself and please ask the question. Nigat Nazir,
So you can unmute, sir. You can ask directly.
आप अपना माइक को अनम्यूट करके आप पूछ सकते हो सर. नो सर, लेट्स गो.
Sir is coming.
No sir, we'll go sir. No.
Okay, I can go for another question.
Yes,
So
please.
this one, Susita, what is the safest level concentration of porphyrin in mushroom?
What is the?
The safe level concentration of porphyrin in mushrooms. I don't know. I guess she is mentioning about the mushroom contains any porphyrins. No?
Actually, whenever you have a porphyrin, okay, that will be colored. Okay. I don't think actually mushroom has a porphyrin in it. Okay, so actually porphyrins are very safe, biodegradable. There is no problem. You know, porphyrin degrades. It will form bilirubin. Bilirubin can be excreted through urine.
Okay, now thank you. Now, so another one question from Pallavi Pandani. So again, Madam asked, how did you synthesize single-walled carbon nanotubes conjugated for firing?
Actually, I showed you the around like here you can see we have prepared monoamino porphyrin. Okay, then carboxy appended SWCNT. Then we can do DCC coupling. So that is the very famous coupling to make amide bonds. So use DCC, we can couple amine with the carboxylic acid, it will give you amide. So amide linkage we have done.
Okay, now thank you. Now, then another one question from Magru Malik. He asked, does increase the concentration of porphyrin polymer leads to decrease the size of nanoparticles?
Actually, for porphyrin concentration, if you increase, then there will be more aggregation. I don't know, it will decrease the size. Okay, you have to have optimum concentration and optimum condition. Like what you should do is that you can use some surfactant, like so, and then we have to do a high speed spinning. Okay, then you can get porphyrin through supramolecular interaction only. It will form J aggregates or H aggregates, so that porphyrin nanoparticle will form. But don't use high concentration, you will get aggregates, larger aggregates.
Okay, now thank you. Now, then another one question from Sasak Sarma. Sir, please give a photodynamic therapy applications. So can you please account some details about photodynamic applications?
Okay. Actually, photodynamic therapy is nothing like you see here. What we do is that we excite porphyrin, it goes to the exciter state, okay, and then there is an intersystem crossing from singlet to triplet. That triplet is lower in energy than singlet as usual. The triplet level will do a energy transfer to a triplet oxygen to become singlet oxygen, or electron transfer to generate superoxide radical, hydroxide radical. They are very reactive. They will react on the cancerous cells. They will kill the cancer cells. That's how that they have done a lot of treatment. Here, you see this chlorine. Why they use chlorine means chlorine has intense absorption in the red region or beyond that. So they will act as a good photosensitizer for photodynamic therapy. They have treated wide variety of cancers like lung cancer, esophagus, okay, and then skin cancer. What he wants, like I'm not getting Pasimale, like what he
Yeah,
wants.
yes, sir. So he wants to know about the photodynamic therapy application. Whichever you given, that is the correct answer. Na? So
Yeah,
I
yeah,
guess it
okay.
is
Thank you,
enough
thank
for
you,
them.
thank you.
And one more question from our students, research scholars from BS Abdur Crescent Institute of Science and Technology, Gokul Ishwaran. He asked about, sir, you have discussed H aggregation and J aggregation of porphyrin.
Yeah.
What way we can distinguish these two? For example, if you are taking TEM image or SEM image, only we are getting aggregated particles. But how we can distinguish this one is the J aggregation, this one is H aggregation?
Actually, UV visible spectroscopy is a good technique. You can identify whether it forms a H aggregate or J aggregates actually. Okay, because these aggregation will give you spectral shifts and then pattern will be different. That.
Okay. Now. And now I request the participants, still you have a chance, you just unmute your mic and you can ask directly or you can talk with our chief guest directly. We have a lot of questions from chatting box. So now I am giving the opportunity. You can directly unmute your mic, talk to our chief guest. Venkat, hand raise, anything it's there? Please check it.
No, sir, no hand raise. No.
Okay. So then one question from Kotit. Sir, my question, what are the factors affect ED on quantum yield?
Okay, factors can affect quantum yield is that first is concentration. Actually, if you keep higher concentration, okay, then that will undergo aggregation. So you will lose whatever the photon through molecular aggregation, like it will be. So you have to use very dilute concentration. Anybody does fluorescence measurements, they know. In case of porphyrins, we keep the absorbance of Soret band 0.1 absorbance value, because you have to take 10 to the power minus 7 molar solution, then only you can disperse the molecules so that the molecular aggregation will not be there. So concentration is the main factor. Okay. After that, like a temperature, other factors can be there.
Okay, now thank you. Now. And Dr. Anandan sir asked about nowadays we are called DSSCs, perovskite solar cell. But is there any new type of solar cells recent days is available?
Yes, recent say I said now perovskite solar cells. That I did not discuss due to lack of time. The perovskite solar cells there, they use perovskite as a light absorber. Okay, then porphyrins are used as a whole transporting material. That means it will give electrons. Then there is a positive charge on the porphyrin that will move. The electrons will move to electron accepting layer. There is a perovskite solar cell. That is a very emerging area. People have observed 22% efficiency. Uh, and there is only one problem is the stability of the perovskite solar cells. So they are very sensitive to moisture as well as temperature, and also that when you keep it in open air, it degrades like anything.
Okay, now thank you. Now. And this question from our department research scholar Chandru, he asked about, sir, you applied the porphyrin for biosensor as well as toxic chemical sensor application. But when we are going for biosensor application, the solubility in water, it is important. But majority of the porphyrin compound, it is not in a soluble, soluble in water. So can you comment on it?
Yes, actually, really, if you want to go completely in vivo studies, you have to just sulfonate it. Okay, so if you stir porphyrin with sulfuric acid to some optimum condition, then you will get sulfonated porphyrin. So sulfonated porphyrins are water soluble. You can use it. They were used as a photodynamic therapeutic agent as well as sensors, biosensors. That so whatever we have done is that it is like in vitro studies. In vitro studies, this chemical sensing for water and other things, we did not do sulfonation. When we want to use it for in vivo applications, of course, we can make it water soluble by sulfonating. Otherwise, also 5% DMSO or DMF solutions, these porphyrins are soluble in water because you need very low concentration of porphyrin. It doesn't require very high concentration. So porphyrin has a moderate solubility in DMSO water mixture, DMF water mixture, or sometimes acetonitrile water mixture to some extent it mixes.
Okay, now what about the organic buffer solution? Can we use that organic buffer for this biological application?
Actually, yeah, you can use PBS for
Yeah.
water. Organic buffer also you can use it. Not a problem.
Okay. Now. And this question from Swapana Narayana Swami. Madam asked about how can we measure singlet oxygen and how can we enhance its quantity?
Yeah, actually, singlet oxygen measurements, there is a specific, like they follow by fluorescence titration. Okay, so they have a system that called fluorescent quenchers. Like, so you, whenever you have singlet oxygen, then when you take acceptor, that will react, then your fluorescent intensity will come down. The fluorescent intensity that much comes down, that we say that it is 60% singlet oxygen production, 80%, 100%. So there is a titration we can do with the fluorophore. The intensity, because singlet oxygen will quench the fluorescence of a fluorophore. So fluorescent titration will tell you how much singlet oxygen generated. So this is an indirect method people are widely using.
Okay, thank you. Now. And
And
another
another
question,
question is that that we
can I
can
ask now?
enhance, we can enhance means we can
Yeah,
enhance
please.
singlet oxygen generation by actually tuning the triplet level. You bring triplet level lower and also your singlet level lower, so that more number of molecule will go to excited state, then it will go to triplet state, that then it will react with oxygen. So that's why we always interested in redshifting the spectrum. When you go more redshift, then excitation becomes easier. More number of molecule will go to the excited state, then more singlet generation will be there. That's why pi conjugated systems are good for oxygen generation, singlet oxygen.
Okay, now thank you. Now this question from Ibrahim, our MSc student, he asked
Okay.
about generally the organic compounds having photobleaching nature. You said that porphyrin also having fluorescence. What about the photobleaching nature of this porphyrin, sir?
Yeah, some of the titanium porphyrins were used for photo bleaching. It can act as a like semiconductor for photo bleaching, but very less number of studies are there as a photo bleaching, because sometimes when you shine light, porphyrin also can undergo photochemical reaction. Okay, so but not like say other organic dyes, under photochemical condition it bleaches, but porphyrin it does not bleach that easily. But if when you use intense radiation for several hours, several days, then they may undergo reactions. But we like I did not see much report on using porphyrins for photo bleaching.
Okay, thanks a lot. Now again, I can give the opportunity to the participant. You just unmute and ask the question to our chief guest. Otherwise, we have a big questions, lot of large number of questions from chatting box. So I can give now opportunity. Anybody is interested to talk with our chief guest? Bring it, Parpa. Just look. Anybody is raising hand?
Yes, sir. Yes, sir.
Maybe it may be the problem for unmute. So if they are
No,
raising
sir. I
hand,
have
just
given the option, sir. I have given the
okay, fine. So participants, we have given the option to unmute by yourself. So you can directly unmute it and talk with our chief guest. Okay, a few more questions is there. If you don't mind, can I ask now?
No problem. Please go ahead. Go ahead.
Yes, no, thank you. No. And this one from Srinisha. What is the
Yeah.
expected mechanism of amino acid based metal complex with porphyrin in NLO applications?
Actually, you have to append peptides, okay, along with the porphyrin, because they have a chirality, okay, that depends on the amino acid you use. So that can be used in chiral application. Right.
And
What is the
Debakanta,
question? Can you repeat once more?
yes, no. What is the expected mechanism of amino acid based metal complex with porphyrin? Amino acid
Indian
based metal
Institute of
complex with porphyrin
Technology, Roorkee.
Yeah,
in
actually,
LLO
amino
applications.
speaking.
amino acid, if you append, then actually porphyrin also will bind with the metal. So if you have to protect the core, then you have to allow to the periphery to bind. then will be the same mechanism, photosensitization, and then it will generate singlet oxygen that will be used for anti-cancer therapy. Or NLO,
Okay.
NLO means like what you are doing is that two photon absorption, but amino acid you can append extra metal for NLO application. Porphyrin also will participate in that. I don't see much like how we can tune the system. Yes. Maybe
Okay, thank you.
give chirality, that chiral nature can help.
Yeah, sure. Thank you. So Venkat, anybody is waiting to talk with our chief guest?
No, sir. No, sir. No.
Okay. Then
Sir,
I can.
myself, I
Yes,
just
yes,
have a
yes. You
small
can ask,
question.
Aisha. Yes.
Nothing, sir. Out of curiosity, I'm asking. Sir, it was a very good presentation. And regarding the photodynamic therapy in cancer treatment, how far is it applied, sir? Can it be an alternative for the gamma radiation radiotherapy?
See, thank you for nice question. Like photodynamic therapy is widely used for skin cancer. It is very easy to use and site specific. You can go to the particular place and you can do the treatment. But the gamma radiation is ionizing radiation. It will give you a lot of side effects. But photodynamic therapy has less side effect. Okay, because you are going to particular, you are targeting a particular place. So this size specific can be there. Then wherever that you need, that place only you will be treating, so that whole body is not exposed to light, or it is not getting sensitive to the light. So compared to gamma radiation, photodynamic therapy is very good technique.
Thank you, sir,
Okay.
for answering the question.
It is
Okay,
widely
thank
used
you.
in US, Europe, or many countries. Only thing is that currently a little bit expensive this technique is, because that porphyrin photophyrin synthesis is little bit tough, then cost is higher.
Okay,
Thank you,
thank you.
sir.
And it's my question. So photodynamic therapy and photothermal therapy, which one is better now? So can we able to use this porphyrin for photothermal therapy?
Yes, 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 we are working on photothermal therapy. Okay,
Okay.
that is actually here your singlet oxygen generation, there your heat generation. Okay, so
Yeah.
actually, in my personal opinion, this photodynamic therapy we like, that is better actually. But porphyrin also can be used for photothermal therapy. There are reports are there. You can use it. That you just you are generating heat, then that kills cancer cells.
Yeah, by using laser, it will kill the cancers.
Yes, yes, yeah. That is the excessive generation of heat there, then it kills there. Actually, they are
Okay.
interrelated. Actually, whatever that you are using for photodynamic therapy, with slight modification, photothermal therapy also can be done.
Okay, now. So thanks a lot for your excellent presentation. And on behalf of Department of Chemistry, BS Abdur, Crescent Institute of Science and Technology, and myself,
Dr.
it is our. Yes, yes, sir. Yes, sir.
1 minute. Okay. Sir, thank you, sir, Dr. Sankar,
Yes, please.
for the elaborate information on porphyrinoids.
Sir, 1 minute, sir. Iswar, sir. I can introduce you. So, Dr. Iswaramurthy is our head of the department. Now he wants to talk to you, sir.
Very nice.
Sir,
Good evening,
thank you,
sir.
sir. Thank you for your elaborate information on application of porphyrinoids and accepting our request also, sir. Thank you.
Thank you, sir. Thank you so much. You're most welcome.
Thanks a lot, sir. Iswar, sir. On behalf of Department of Chemistry, BS Abdur, Crescent Institute of Science and Technology, and myself, thanks a lot, Anna. Thanks for your kind acceptance and come over and deliver a talk during your pandemic period. I know well you are very busy during your busy schedule. You accepted our invitation and the talk was very excellent. We fortunately have 1,500 registrations. Many people have seen your talk as well as live stream. YouTube also is going on. We also kept this YouTube for a few more months. We are going to keep. So many people will see. 3 days over on YouTube. We just telecast it, and we got 1K views. So that much things is going on. So different categories of participants we have received, from the director principal, HOD, and multidisciplinary areas participants we have received, as well as our participants from throughout the India, Meghalaya, Sikkim, Assam, up to all the areas we have it. So thanks a lot for your kind acceptance and come over and deliver a talk. Now I hand over the session to Aisha.
Thanks, Vasimale.
You're welcome, ma'am.
Thank you, sir. Thank you, sir. It was a wonderful lecture. Thank you for sharing your valuable insights and your precious time. You have covered briefly and very elaborately on solar cells, photodynamic therapy, mechanism of photodynamic process, treatment of cancer using these porphyrins, chlorins, and antiviral activity, and the materialistic applications of porphyrinoids in different fields. Thank you, sir. I'm sure it was an eye-opener for everyone. And now I take wholeheartedly, I thank everyone for organizing this event. And now I'm extremely happy and privileged to have been asked to propose the vote of thanks. First and foremost, I would like to thank the speaker, Dr. M. Shankar sir, who has shared his valuable time with us today and made us to get benefit from this session. And I thank you very much, sir, and I extend my deep sense of gratitude to the conveners of this program, Dr. S. Kutirani ma'am, Professor and Dean, School of Physical and Chemical Sciences, and to our beloved Professor and Head, Department of Chemistry, Dr. D. Ishamurthy, sir. I would also like to acknowledge and thank the coordinator of this FDP program, Dr. N. Vasimalai, sir, for his sincere efforts in making this FDP program successful. Last but not the least, I would like to appreciate and thank the audience for their extreme cooperation and support, even during the most challenging time of humanity, the COVID-19 pandemic. Your active participation and commitment keeps our spirits and hopes up. I once again thank you one and all, and I wish you a fruitful and happy evening. Stay home, stay safe, and take care. Thank you, sir. And
Thank you,
thank you
ma.
for giving me the opportunity to host. Thank you.
Thank you, ma. Rani madam, Dean madam, are you there? Madam, if you're there, just unmute and talk with our chief guest, madam. Vengat madam irukangala parpa?
No, sir, no.
Okay, sir.
Thank you, Vasimale. Thank you so much. I'm
Yes, sir. Thank you so much, na. Thanks a lot for your excellent talk, na.
okay, sure.